Thank you very much, Chrissy. My name is Cipriano Zaveno. I'm one of the two conveners of the Marine Artificial Intelligence Forum of MASTS. Like all MASTS forums, we are designed as a medium for knowledge exchange, education, and networking. And yes, as you might guess from our name, what we're particularly interested in is trying to contribute to advancing AI-predicated solutions that target specific problems in the marine environment. This usually includes state-of-the-art AI techniques that exist in research, but also developing new techniques, or specializing them to the particularities of the problems in the marine space. We have today with us a very interesting presentation, Dr. Cameron Trotter from the British Antarctic Survey, that's going to give very interesting insight on how deep computer vision, so machine learning, in the computer vision domain has been used to understand and to, I would say, better quantify at speed the current state of the benthic ecosystem in the Southern Ocean. So yeah, without further ado, Dr. Cameron Trotter.
Thanks, Cyprian. Yeah, hi everyone. It's good to be here. So yeah, I'm Cameron and I'm a machine learning research scientist at the British Antarctic Survey. And I'm going to talk a bit about our work and the challenges that we faced, I guess, with trying to use deep learning and computer vision to understand the current state of Southern Ocean benthic or seafloor ecosystems. So Southern Ocean ecosystems are most known for their higher order predators, such as seals, whales, or penguins, as well as their abundant plankton life, things like krill and copepods. However, the highest biodiversity in the Southern Ocean ecosystems is actually found on the seafloor, the benthos. And yet this is a relatively understudied area given its remoteness, as well as its difficulty to access. So for reference, approximately 70% of benthic sampling sites around Antarctica are within 350 kilometers of a research base, and those are shown by the dark points on the map there. So that leaves large areas that have not been studied well or studied at all. So I'm part of a larger project aiming to understand the evolution of these ecosystems. So using the fossil record to try and understand the past, and then biodiversity monitoring to understand the present, and then use the information that we gain from that to understand how these ecosystems have changed over time. So from a temperate rainforest environment to what Antarctica was millions of years ago to what we know now. And the idea is hopefully we can then use that to model how we expect the environment to change as it warms back up, and what effect this will have on the Southern Ocean benthic ecosystems around Antarctica. So for most of human history, our understanding of the benthos has come from nets or trawls. And these have advantages, and they're even still required for some things. So like if you want to do DNA sequencing on an organism, for example, you're going to have to bring it up. But it can be overkill for some monitoring. So trawls are inherently destructive because you're physically removing the organism from their ecosystem, and you also might not get everything. So for example, if something is particularly hardy and is stuck to a rock, or is fast enough and can get out of the way. And you also lose some structural information because everything is brought up in the net in one go, so you don't have any idea where things were, what they were next to, things like that. So towed camera systems like the OFOB system developed by the Alfred Wagner Institute, who are the German Polar Research Institute, and the OFOB system is that camera system that you can see there on the bottom right, allow for in situ monitoring over a very large scale. So essentially like a net, you throw the camera off the side of the ship, you drag it along and it sits in the water column and kind of just bobs along the seafloor, taking photographs as it goes. And so this is where the data from our project comes from. So RV collect that data, and then BAS ecologists will label that data and try and identify all of the organisms that the camera detects on the seafloor or captures on the seafloor, I guess. And we're currently focusing on two cruises in the West Antarctic Peninsula, which is the little spiky bit that sticks up kind of towards South America. So this is an example image that you would get back from the OFOB system. This is scaled down to fit on the screen. They're very high resolution. They're about 8,000 by 5,000 pixel. And they're extremely noisy. So there's a lot of different environmental conditions, a lot going on. And we also have very high organism abundance, like I said, because of the remoteness, but also in part due to protection policies, so things like the Antarctic Treaty System. So labeling that high resolution imagery can be an extremely lengthy process. So current processors use Inkscape, which is essentially an open source version of Photoshop, to draw around organisms directly on the imagery. And every layer in that Inkscape image contains a category of organism. And these can be very fuzzy. So for example, we've got strawberries, question mark, chunky white star, worm, question mark, and it might also contain other auxiliary information as well. So for example, the percentage of encrusting that's found in the image. So labeling this up manually requires a very high level of taxonomic expertise, especially given the levels of endemic organisms that are found in the Southern Ocean, and also just the abundance as well. So we have about 100 images labeled at the minute, all done by hand. And that takes on average about 8 hours per image in order to fully label. And we currently have a backlog of about 33,000 images. So yeah, if anybody wants to spend their life labeling up this imagery, let me know. And that kind of motivated this question, I guess, of can we start trying to automate some of this data curation using computer vision? And I guess kind of that's where I was brought into the project. And the answer, I guess, is mostly yes. So this is our methodology, and I'm going to go through the specifics of each step, but I thought it would be good just to kind of give you a bit of an overview before we go in. So like I said, we have 100 labeled OFOBS images, and they're all in Inkscape. So I spent a lot of time converting them from Inkscape SVG files into something that's actually usable for computer vision. I listened to 6 audiobooks while I did this. I reckon I could probably go on Mastermind, and like my topic of choice would be the Inkscape, the SVG file format documentation. I also spent a lot of time with ecologists at BAS, kind of condensing the fuzzy labels that we have down into about 25 different morphotypes based on visual distinctiveness, and they became our object detection classes. Some of the imagery, parts of it wasn't also fully labeled. So for example, you might get parts of the edges where distortion has occurred. So I cropped those down to help with class confusion. So we have a very small subset of available images that are labeled, and we're biased, I guess, for us towards relatively shallow depths of around about 500 meters, as well as 1,500 meters and below. The labeled imagery is also biased in its seafloor angle. So about 52% of those images that we have labeled are captured on a slope of less than or equal to 10 degrees. The data is also massively skewed towards soft substrate images. So about 61% of those are captured in muddy environments. But the number of organisms in the hard substrate or rocky environments massively outweighs the soft. So about 95% of our annotations are on hard substrate imagery. So when it came to deciding on things like the data set split, rather than randomly splitting those images into our train, validation, and test sets for building our object detector, I did this based on the annotations, taking into account the substrate differences. So this initially gave a 75%, 15, and 10% train, validation, and test split, which then I modified to account for the depth and seafloor angle distributions, trying to ensure that we had a representative mix of each per split. So this gave, I guess, a rather unconventional train validation and test split of about 69, 19, and 12% respectively. But you can see there, like on the side, the number of annotations that we have in that are massive for especially for those 100 images. Like, you know, our training set has about 69 images in and has 21,500 annotations. The imagery is also extremely high resolution, and it's a bit too high resolution for standard object detection models. So naive approaches like, I guess, kind of resizing here would prevent the model from learning correctly, because we have a lot of very, very small objects, and resizing that imagery down removes all of their features, and it stops the model from learning about them. So we've adopted a methodology called SAHI, which stands for Sliced Aided Hyperinference, I think. And essentially what we do is we take our high resolution image and we run a sliding window over, and we generate patches of fixed size from those high res imagery. And then we train our object detector on those patches. And what that allows us to do is we get the detections on a patch, and then we reposition those detections onto their location in the high resolution imagery. And we also generate our patches with some overlap. So we do some post-processing to remove multiple detections of the same object. And this allows us to keep kind of the high resolution per patch whilst keeping a consistent image size. Because I said, if you remember before, we had to crop some of that labeled imagery down. So in order to use SAHI, there's a lot of, I guess, kind of hyperparameters that you have to deal with or set. So to figure out the best for our project, I started by essentially just training a lot of kind of baseline faster RCNN models for each different combination of patch size, overlap, and minimum visibility. And when I say minimum visibility, what I mean is how much of a box should be left if it's split between patches in order for that box to be considered a valid ground truth. So for our data, the best combination we got was 500 by 500 pixel patches with a 50% overlap and a 25% minimum visibility. And this gave us about 25,000 patches to use for model development. And so we use that kind of going forward. I also examined the optimal hyperparameters for the SAHI post-processing. So SAHI has two main merging algorithms, non-max suppression. So if two boxes overlap, retain only the most confident, and non-max merging. So if the boxes overlap, merge them together. And we found that non-max merging was best. Also examined the optimal match threshold. So what percentage overlap should there be between boxes before we start merging them? And this came out at about 20% for our use case. And I trained lots of different models here for this, and given time constraints and also the fact that it would just be really, really boring. I'm not going to show you the metrics for all of them, but I will show you the metrics for the models later on. One thing I probably should mention, all of the models in this project were trained using the detection framework, which is available on GitHub. And I found it really useful for very quickly kind of training up a lot of different models. Also did some data augmentation stuff. We found spatial level was best here. So kind of flipping, doing random cropping, that sort of stuff. We found pixel level wasn't so great. So adding extra motion blur, random shadows, changing the brightness and contrast. And I guess my gut feeling on this is because our camera system is already moving anyway, we're already seeing a lot of that type of variation. So adding more on top is more of a hindrance than a help. I also looked at different architectures. Sorry, I really hate this slide, but it was the only way I could convey the information. If you really want to read the table, look at the averages at the end. But generally, I guess kind of what we see in the evaluation metrics, at least for version 1 of our model, is that they aren't so great. But these are very, very noisy images. It's a very, very difficult detection problem. And we also only have 100 images. So I examined the effect of pre-training on the MS COCO dataset, and we found that it helped, but not by much. And there's also very little difference between CNN and transformer architectures here. Transformer architectures are more computationally expensive. So are they worth it for us? Maybe, maybe not. And even with using that SAHI post-processing, we're finding that these standard architectures are really struggling with small objects, which is most of our objects. So we divided our objects into small, medium, and large based on bounding box area. And what we're seeing is that no single model architecture that we've tried works well for all sizes. So we might need to look at model ensembling as well. So using a different model to detect small, medium, and large things. I would also want to point out that I guess those first 3 points on the slide, they might not be so much of a problem as we start clearing that data backlog and we start kind of generating more data. It might just be a data volume problem, I guess, rather than anything else. I really want to talk about the effect of abundance of organisms on the model performance. So this is the confusion matrix for the best performing model that we have at the minute. And this is ordered by abundance. So stylastrids in the top corner there are our most common, all the way down to ascidians, which are our least common. And abundance is significantly impacting model performance, and I think it affects it more than object size. So for example, stylastrids are small objects, and ophiuroid five arms, which is a few down and along from stylastrids, are large objects. And what we're seeing is that relative to other classes, we're seeing fairly good hit rates, especially compared to our rarer organisms that are at the extremes. But they have, in some cases, less than 100 examples in our data set, and we're getting basically a 0% hit rate on those. But what I think is interesting and is good from our perspective is that kind of that final column there, the background column, is absorbing a high percentage of these misclassifications. And essentially what that means is that our rare things are often missed by the model rather than mislabeled. And that's good because essentially we're not trying to replace the ecologist here. We're trying to provide them a tool to reduce their workload. And so what we can gain from this is that if our model produces a box, chances are that box is labeled correctly. And so the ecologist essentially just needs to fill in the blanks rather than kind of starting from scratch. So I guess this is as high res an output as I can give you on a PowerPoint presentation slide. So this is back to 8,000 by 5,000 pixel. And you can see we're starting to detect quite a lot of stuff there. There's still a lot of things that we're missing just because our abundance is so high. But like I said, this allows us to do a first pass before the ecologists see it and massively reduce their workload because they're just filling in. They're not having to look at all of the image. So that's our methodology. Again, so like I said, we take those 100 labeled OFOBS images, we split them based on substrate depth and seaflow angle to generate our splits. We then patch those up and use them to train an object detection model. And then we use Sahi post-processing to recombine those detections onto the high-res image. And what that allows us to do is take those unlabeled high-resolution images, run them through our object detector, and generate predictions without losing any resolution. So future plans. So like I said, this is kind of version 1 of our model. And what we really want to do is kind of start looking at an active learning or human in the loop scenario here. So what I mean by that is, so we have our version 1 model, which is trained on 100 images. Let's take another 100 unlabeled images and let's run those through our model. That'll generate some predictions, and then our ecologists will correct those predictions and fill in the blanks. Now we have 200 labeled images, which we can then use to train version 2 of our model. But at the same time, we've also cleared some of that backlog. So now we can use version 2 of our model to feed some more imagery, unlabeled imagery through, clearing the backlog further, but also retraining our model whilst reducing the amount of time that the ecologists are having to manually curate that data. Because what you would expect is as the model performance improves and we get more data, the amount of time that the ecologists are having to spend is coming down. Another interesting thing is kind of looking at post-processing failures, especially for large objects. So like I said, Sahi is primarily designed for satellite imagery, and that expects a consistent object size within classes. And that's not really the case for our data because our camera is free floating. So objects could be close to the camera or far away from the camera. And this leads to some post-processing failures, especially for large objects that are split over a lot of patches, like the ophiuroid that you can see on screen. And long term, we want to look at kind of spatiotemporal and sensor changes. So how this methodology can adapt to handle new environments and new camera systems. So yeah, that's what I've done so far. I hope it's of interest.
I think it is. It's very, very
Good.
interesting. Thank you very much. So it's a great talk.